with us. And we can see your
Great,
slides.
Yeah. Great. So yeah, thanks for everyone attending, and I'm happy to be a part of this presentation here. So as Allison introduced me, I'm a staff scientist here at EMSL, working with a great group of biologists and environmental scientists. And our work is focused on interrogating interactions and absorption of organic carbon to mineral surfaces using some novel nanometer techniques to achieve high sensitivity and high spatial resolution. Okay, there we go. So the motivation of our work is to understand interactions between organic carbon and minerals, in particular in soil environments. And so the overarching goal is to really get at and understand from a mechanistic perspective the persistence of organic carbon in soil environments. And this is particularly interesting because organic carbons in soils exceeds by some estimates the amount of organic carbon in the atmosphere and vegetation pools combined. And we know that this terrestrial organic carbon is particularly sensitive to decomposition due to climate change with increasing temperatures. So to understand its fate in a changing environment is very important. And it's well understood that its stability really hinges on its interaction between various minerals in the environment. And so our goal of this research is to understand from nanoscale and mechanistic perspective these interactions and what governs the stability of this organic material in the soil environment. This cartoon illustrates our experimental approach. So we have classes of organic materials that we react with mineral surfaces. The organic materials are selected from the four classes of canonical materials, including lignin, fatty acids, amino acids, and carbohydrates. These are reacted with calcium carbonate nanocrystals, and we study their absorption and stability using imaging and spectroscopy techniques. So our first approach is to really image the nanoscale formation of and nanoscale behaviors of these materials through electron microscopy. And this shows its transition from the dissolved organic materials and the pristine crystals to insoluble organic materials that nucleate from these facets and edges of the calcium carbonate where the surface energies are much higher. So the SEM images show that the organic material is localized on these mineral surfaces and have features that extend from microns down to tens of nanometers or below or lower spatial features. And we see that this is highly dependent and it arises a lot from the specific mineral configuration. So what's missing from these maps are the organic material identity the SEM can only provide morphological information. But to understand this in more depth, we need the help of chemically specific techniques. And this is where a new technique comes into play. It's called scattering scanning near field optical microscopy. And it is performed as a modification of an atomic force microscope. Briefly, this operates by bringing a sharp tip to a sample surface, and it's scanned across to collect topographic information. To implement the chemical identification and spectroscopic capability, we then illuminate the AFM tip with infrared or terahertz radiation. This infrared light then is localized and concentrated by the AFM tip, and can actually interrogate the sample chemical information with very high spatial resolution. And this has been widely successful in biological fields, showing down to single protein complex sensitivity. Here, this is a map of a ferritin protein complex, where we see that the AFM topography shows a single protein complex on a surface, and its map of the infrared absorption of its amide band shows a clear localization of the protein. This has also been shown to map the concentration of membrane protein in Bacteriorhodopsin, where we have a membrane protein patch on a surface, and we can map this protein content with down to 20 nanometer spatial resolution. In tandem with this, we can also get spectroscopic information by sweeping the wavelength, and it shows that you can get the amide band of the protein well resolved with down to 10 nanometer spatial resolution. So a bit more about the experimental apparatus and setup. Again, it's based on atomic force microscopy, where a sharp tip is scanned across the surface. Our source is an infrared laser, which is tuned to the vibrational resonances of the target material. We use an interferometric detection akin to FTIR, then to get the spectral information. It's, as I said, it's particularly well tuned to get at protein and amide signatures. Here is an image. Here's an image of the organics dispersed on embedded in the nacre region of a mollusk shell. So the cross section shows that we have thin organic lines related to how the oyster grows, and can tell us information about its about the role of proteins in the shell growth. I'll also highlight that there's a large dynamic range with this technique, where we're imaging features that are 50 nanometers across over tens of micron spatial extent. And so you can really capture these multi-scale images using this technique. And again, to confirm the chemical identity, we can perform point spectroscopy by localizing at one point and getting that FTIR spectroscopy information. I'll also highlight that this technique can be, since it is non-destructive and minimally invasive, it can be correlated with other techniques. Here we're correlating with nanoscale secondary ion mass spectroscopy. This is proven to give very high spatial resolution of elemental information. Here we have selected a number of different organic compounds to map. And what we see is we also have this well-correlated image with the infrared spectroscopy technique. And so this gives complementary information for these elemental techniques to actually give that chemical amplific response. So pivoting to the core of this study, we have focused on studying the absorption of organics onto mineral surfaces. So here is a demonstration of one of the experiments. Here we have the AFM height of a hematite surface that's exposed to an organic solution. We see various adsorbates on the mineral surface. And in our infrared map, we see that there's specific locations that are that show this peak at 1740 inverse centimeters. And this corresponds to CO double bond stretch, the carbonyl. And this shows that we can get that chemically specific information about the adsorbates with very high spatial resolution. So in the core of this study, we studied these calcite nanocrystals that are reacted with organic mixtures, and they show this hierarchy of nanoscale structures in the SEM. And our goal is to get the chemical information of these adsorbates and these structures without compromising spatial resolution. The organic compounds we've selected, again, are from 4 classes of these canonical materials. Specifically, we look at pentaglycine, lauric acid, trehalose, and lignin. Our vibrational spectra guide our experiments. So we perform far-field FDIR spectroscopy on these compounds and locate specific peaks of interest that will aid in our identification of these compounds with our nanoscale technique. This image shows a correlation of various different nano imaging techniques. First, we have the SEM to give us a map of the interesting features that we can focus in on with our infrared technique. The AFM height is then recorded, as well as the chemical maps of the feature we identified. We have selected this region due to its nice combination of different morphologies. So we have the ring structure, as well as a thin linear structure here, fibrous structure, as well as a number of other amorphous and non-specific areas. You see that they have these features have generally high amide and aromatic responses. And we can also correlate this with other elemental maps here, provided by X-ray absorption near-edge spectroscopy stains. And we see that we can correlate the identity of these organic materials and the association of the calcium, and gives us insight into the calcium coordination. So focusing in on a few different areas, we looked at this circular region identified in SEM. We looked at performing a map across this. We have a high region in the middle, as well as linear features in the corners. And our infrared map at 1600 show a contrast across this region. And we selected 3-point spectra to try to get at the actual chemical information of this chemical content of this area. So the correlated point spectra are shown in red, green, and blue here. So we see that they have similar spectral features, and we note that the features at 1600 and 1480 correspond to the lignin lignin spectra, which indicates that there could be a high content of lignin in these areas. We also note that there's very little amide 1 and amide 2, which are characteristic of the pentaglycine here. And there's very little carbonyl response, which shows that there's not much integration of the lauric acid into these compounds. So now focusing in on a ring structure here. We have again a map at 1600 and a point spectra at on this ring, which has very similar spectral features with very low amide bands and very little carbonyl, with the predominant spectral features originating from the lignin, which again suggests that these ring-like structures that form are primarily composed of lignin. So all these experiments that I just mentioned, they're all done ex situ after the reaction. And what we're moving into now is to try to do these experiments in situ using a new technique that's been recently developed in EMSL. So this technique is a way to do in liquid infrared S-SNOM using a different geometry, borrowing from a total internal reflection spectroscopy. The schematic is shown here. We have an infrared source that is specifically tuned to the vibrational resonance that we are targeting. This light is focused using an off-axis parabolic mirror through a zinc selenide prism. This prism is used in total internal reflection geometry and has this evanescent field that decays into the sample surface, the aqueous sample surface. And the AFM tip then probes this infrared response of the material, which is collected and used for interferometric detection. So this evanescent wave then excites the sample and provides low water absorption and low far field background, as well as having this additional benefit that there's enhanced interactional emission of our vibrational signatures via tunneling of high wave vector photons. And we have very high spatial confinement and thereby very high molecular sensitivity. So as a test demonstration, we have imaged the absorption of proteins on a metal surface. Metal surfaces in particular work very well for our measurements because it has very high infrared conductivity and can give us very high spectral sensitivity. So here is a gold triangle on our zinc selenide surface with protein on the surface, and we have taken point spectra across this dashed line here, here shown as in the vertical axis. So on the right we have the protein, and on the left we have the gold surface. So the amide signature shows very strong on the protein, and we can resolve the location of the protein with down to 50 nanometer spatial resolution. So this demonstrates that we can actually get this high chemical and spatial sensitivity performed in liquid environment. So as an application to a natural protein in its native aqueous environment, we imaged and did spectroscopy on a crystallized catalase structure. So this is a protein with complex folding and secondary structures. And our aim is to map and identify the nature of this system. So the catalase structure is raised and on the right here, and we did a point spectra on this, and the amide response consists of and the broad amide response can be deconvoluted into 3 different conformational structures of the protein. So
1 minute left.
The beta. What's that?
About 1 minute left.
Okay, thanks. We have the beta sheet, alpha helix, and random coil as expected. So this brings me to my outlook. So with our new in-liquid technique, we can access a wide range of different new problems in environmental and biological science, from conformational changes in proteins with change in pH or temperature, as well as the in situ real-time detection of organic and inorganic adsorption and interactions, as well as ex situ biomineralization, as shown in the organic maps in the nacre regions. So with that, I'll thank you for your attention and take any questions.
Thanks so much, Brian. That was a really great talk and fascinating research. We don't have time for questions right now, but people can still post in the chat or go